Good morning and welcome to Informa Pharma Intelligence's post-ASCO webinar. We have a team of expert analysts who are on the ground in Chicago ready to share their insights on the key developments, takeaways, and implications from the conference. And we're excited today to have key opinion leader Peter Lee from City of Hope joining us. I'd like to start by thanking everyone for taking the time to join today's webinar and run through some quick housekeeping items. Today's webinar is being recorded, and everyone will receive an email with a link to view a recording of today's event. You are invited to submit questions during the discussion. You can use the control panel on the right side of your screen. Toward the bottom of that panel is the Q&A pane. Submit whatever you'd like. We'll try to work your questions into the discussion, or we'll follow up after the webinar by email. Now let me introduce the panel. My name is Mary Jo Laffler, and I will be your moderator today. I head up the commercial content teams for Script and the Pink Sheet, where I've spent the last 15 years reporting on the pharmaceutical industry. I'm joined by Dr. Peter Lee, Chair of the Department of Immuno-Oncology at City of Hope Comprehensive Cancer Center. He also serves as co-leader of the Cancer Immunotherapeutics Program and a professor in hematology and hematopoietic cell transplantation at City of Hope. His research focuses on understanding how cancer impacts host immune responses in patients, with the goal of developing novel treatments to restore and enhance immune function in cancer patients. His group aims to dissect the complex interplay between immune stromal cells and cancer cells within tumors, tumor-draining lymph nodes, and blood using experimental and systems biology approaches. His group has shown that lymphocytes from patients with breast cancer, melanoma, and colorectal cancer develop immune signaling defects that block their proliferation and function in vivo. He is also extending these findings in multiple directions, including to other human cancers such as prostate cancer and leukemia. Allison Bruce is a Principal Analyst in Oncology and is part of the Citeline team. She has been an analyst with the group for over a decade with experience in oncology, CNS, and analytics. Prior to Citeline, she was a healthcare consultant in Cambridge, Mass. And spent several years coordinating clinical trials in the Psychopharmacology Unit at Tufts Medical School. Zachary McClellan is an analyst with Data Monitor Healthcare, specializing in the coverage of breast and gynecological malignancies. Zach has a master's in science and biotechnology and entrepreneurship from New York University and worked as a chemist prior to joining Informa. Last but not least is Robert Jang, the Senior Scientific Analyst overseeing Biomed Tracker's oncology coverage for the past 5 years. Before joining Informa, Robert was a cell biologist who earned his PhD from Stanford University before working as a postdoctoral researcher at Berkeley and a staff scientist at the Sanford Burnham Institute specializing in cytoskeletal dynamics. Our analysts have all picked some key abstracts to review, and we'll throw in some general discussion too as you send in questions. Now let's get started. ASCO is the world's largest stage for cancer research and helps guide practice and investigation into new directions. Let's start out by talking about what you all think are the most important trends in oncology right now. Dr. Lee, can you get us started?
Hi. The question was the most important trend?
Yeah.
Okay. Yeah, so I've been interested in cancer immunotherapy for 20 years, and it's really amazing to see how the field has finally become accepted and really taken off for the last few years. So I think one of the things that's really exciting beyond the proof of concept that the host immune system can respond to cancer and can play an important role in controlling it is the notion of that the immune response is complex, and most likely doing a single thing to the immune system is not sufficient to drive the immune response all the way through to completion. And so there's a lot of interest in combination immunotherapies, and I think a lot of this discussion will center around different combinations. And so I would say one of the most exciting things is to really understand how to rationally combine and sequence immune modulating agents.
Mm-hmm. Great, and just check in with the rest of our panelists on the same sort of topic, what you guys see as the most exciting things going on right now.
Sure. This is Allison. As Dr. Lee said, I think the combination agents and combination therapy, to me, are the most exciting, and that's what I took away from ASCO. There were a lot of abstracts presented and data shown that this the developments in this area are really promising, and there's a lot to be done, but a lot to look forward to.
Great, thanks. Robert, are you?
Can you hear me now?
Yes, thanks.
Okay, sorry about that. I mean, I agree. I think as someone who's gone to ASCO for the last 5 years can really see how immunotherapies have just completely overtaken the oncology in so many different tumor types. And you see this year how it's already matured to the point where combination therapies are really really the focus now. When I get to my slides, I'll talk a little bit about where we might go outside of immunotherapies in terms of targeted therapies for very specific biomarker-defined subgroups.
Great, and Zachary, do you want to chime in too?
Yeah, thanks, Mary Jo. I agree with everyone, obviously, that immuno-oncology, personalized medicine is the way everything's going and will continue to go. From the breakthroughs of PD-1, PD-L1 inhibitors, CAR-Ts, and all the combinations that are coming up as we go forward. I think something else that we need to look forward to will be what patients will benefit from these treatments the most. And I think this means we need better prognostic, mechanistic biomarkers to better understand who to treat and how they will respond. So I think that's a key theme going forward as well.
Great. Well, certainly from attendance at sessions, it seemed like the early data on combination strategy was of a lot of interest. And Allison, you picked some immunotherapy combinations. Can you walk us through that research and tell us a bit about how the mechanisms work together?
Oh, sure. So the abstracts I'm going to briefly highlight, I believe, are good examples, and as I mentioned a minute ago, of the immuno-oncology combination landscape. And they show the potential benefit of the combination therapy. And here on this slide, the CheckMate 67 trial is a phase 3 trial of nivolumab combined with ipilimumab in treatment-naive patients with advanced melanoma. The combination of these drugs, a PD-L1 antagonist, which is Nivo, and a CTLA-4 inhibitor, IPI, has a potential in many additional tumors, not just melanoma, but others such as non-small cell, renal, ovarian, colorectal. But in this particular study, the CheckMate 67, 945 previously untreated patients were randomized to receive either Nivo plus IPI, Nivo alone, or IPI alone, and the doses shown here on the slide. The patients were stratified by PD-L1 status, BRAF mutation status, and M stage. And the 18-month follow-up, it was, as you can see on the little chart on the right-hand side, the median follow-up was significantly longer for the Nivo and IPI group in the Nivo-only group, but not in the IPI group. I think I just said that wrong. The Nivo-only group, but not the IPI group. And in addition, the overall response rate for the first two groups was significant at 57.6% for the Nivo and IPI and 43.7% for the Nivo alone, but only 19% for the IPI alone group. And then the conclusion note, as you can see here, that the Nivo and IPI and the Nivo alone groups showed significant improvement, improved response in these groups versus IPI alone, regardless of the PD-L1 expression or the BRAF mutation status. But I think that the PFS in the BRAF mutation mutated patients receiving the Nivo and IPI needs to be noted here, and I thought this was particularly interesting because in the study a PFS of 15.4 months was seen in this subset of patients, which is better than that seen in the COMBI-D combination study, which was published, I believe, last year, in dabrafenib, which is a BRAF kinase inhibitor, and trametinib, which is a MEK inhibitor, in BRAF mutated positive patients with melanoma, and for which the FDA granted accelerated approval for that. The PFS in those patients, in the COMBD patients, was 9.3, and again, in this group of Nivo plus IPI group in the Checkmate 67, it was 15.4, so higher. And then a couple of other things I wanted to point out from this trial was that the BRAF positive patients in the Nivo-only group actually did worse than the BRAF wild type. The PFS of a PFS of 5.6 in the BRAF positive patients versus the 7.4 in the wild type. And the overall response rate for the BRAF positive patients was 36.7 and 46.8 for the wild type group. And then just one other thing I wanted to point out from this trial is that, although there was a greater PFS benefit for the Nivo and IPI group relative to the Nivo only, in the low PD-L1 expressors, the ORR in the Nivo and IPI group was significant regardless of the PD-L1 expression, which was at 57.6. So I think this trial highlights, among others that were presented in other trials that are going through development, is the benefit of the immuno-oncology combination therapy, and perhaps I think, as Zach mentioned, I think highlights a need for perhaps better prognostic biomarkers. So hopefully, we'll see more of that, because I think there's a lot of positive evidence that the immuno-oncology combination therapies are beneficial, but that biomarkers, I think, are something that is a big need to stratify these patients.
And
yeah, go ahead.
And then just to sort of to clarify, because the combination of Bristol's Opdivo and Yervoy is approved in the melanoma setting, and they're certainly looking to expand it into lung and other settings. But I think what was interesting here is you were talking about sort of the BRAF mutation status, and I think with some of the other stuff you picked up, it's going to be interesting to see how the immunotherapy fits into the space alongside the targeted therapies.
Yes, yeah. No, I think you're right, and I'm sorry. I'm just trying to think. I was going to try to pull out some statistics in my head, but I'm not. I don't want to mess them up. So perhaps when someone else is talking, I'll get them and come back to that. But no, you're absolutely right. I think it's something to look forward to. All right.
Great. And so I think in your next slide,
yeah,
that was you're pulling in more of the combination just with Merck's Keytruda.
Yes. Yes, and I think this trial, again highlights the potential of the combination therapy. In this case, a triplet combination treatment. The Keynote 22 was a phase 1, or is a phase 1/2 study. It's ongoing, and pembrolizumab in combination with dabrafenib and trametinib for the BRAF mutation-positive patients with advanced melanoma. So this trial was using a triple combination treatment with BRAF, MEK, and anti-PD-1 inhibitors. The phase 1 portion looked at the MTD, which was determined for triple combination, evaluate safety, and it's looking to evaluate preliminary tumor activity. And then the doses used here, as you can see on the slide, in the non-randomized phase 1 portion, dose-finding portion of the study, they started with the approved doses, and I believe that they had planned to I think, change, play with some of the combination. Anyway, this turned out to they ended up that they're using this combination. So this was the MTD, which is shown here, which is the approved levels, and the outcome was it showed a manageable toxicity profile in the first 15 patients treated. But in the phase 2 randomized portion of the study, the patients will be evaluated for safety, tolerability, and then the preliminary efficacy for this triplet combination in first-line therapy. And to me, this is exciting, and as we mentioned a few minutes ago, kind of the combinations of these different agents and what will work. As I referenced in the previous slide, the COMBD demonstrated superior anti-tumor activity when combining the dabrafenib, which is, again, that would be a BRAF inhibitor, plus the trametinib, which is a MEK inhibitor, compared to single agents. And then there have been several studies showing that pembrolizumab has a manageable toxicity profile. And so I think that again, is the need for novel drug treatments and this triplet combination of BRAF, MEK, and PD-1 kind of could prove to be very promising. So if you want to move to the next slide, we can do that.
Sure.
And this is a graph that's compiled from data pulled from Trial Trove, and it shows the number of immuno-oncology triplet combination trials by phase and status. And as you can see, there are about 4 planned trials and 11 open, 1 closed. So the development in this area is definitely moving ahead, and there's a lot to look forward to. Hopefully, very promising data in the future.
Great.
Yeah.
So I was just going to ask where you see immuno-oncology advancing. What are some of the most promising classes and combinations, and then we'll sort of throw it open to the rest of the group to talk about the same thing.
Well, I think, as I just mentioned, I think that there's been solid data showing that the BRAF and MEK combination, the PD-L1 CTLA-4 combination, and then hopefully the Keynote 22, a combination such as like the MEK, BRAF, and PD-L1 combinations will show promising results.
Thanks. And one of the questions, just while I still have you before we switch to the others, is we people asking about what the toxicity profile of the combination was.
For the Keynote 22?
I would say we could maybe talk a little bit about both, about the differences in toxicity. I know that there's a greater rate of toxicity seen when you add ipi to nivolumab in the melanoma setting. Yeah, so maybe the Keynote 22 being sort of the earlier combination.
Sure. So the toxicity profile for the Keynote 22 did show, let's see, there were, I don't want to give you a whole bunch of, I've got the data here.
Why don't we come back to it, and right now just sort of ask Dr. Lee and Robert and Zachary if they want to sort of weigh in on sort of the next big things in immuno-oncology, new classes and combinations. Dr. Lee?
Yeah, well, I can briefly address the toxicity issue. So, yeah, that's a very important issue. So as probably some of you already know, the toxicity with anti-CTLA-4 antibodies is actually significantly more than anti-PD-1. And so there's this number of autoimmune side effects. One of the more worrisome one for IFI is colitis, which can happen fairly early or late in some patients. So when you combine the two, the toxicity is more driven by IFI, but there's also the additive effect as well. So definitely autoimmunity, particularly colitis, are concerns with these combinations, and that's why I want to certainly agree with the point that Zachary and Allison made about selecting patients. Certainly, because of the side effects, one wants to select patients who will benefit from the treatment rather than just develop side effects. And so that's, to me, an extremely important area in the field, and it's one that my own group works on quite a bit. And I want to echo the the theme, well, one of the themes of this discussion of combination, to say that when we think about biomarkers, we should also think about combinations of biomarkers instead of just a single one. So right now, the field primarily uses a single biomarker, which is presence or absence of PD-L1 in tumors. And I think many of us are now already moving beyond a single biomarker to ask what are the combinations of biomarkers that would be more informative to select and predict response.
Dr. Lee, this is Robert. I'd like to follow up on that a little bit and ask you a question about the biomarkers that you're just talking about, because although you mentioned PD-L1 status as one of the pretty much the only biomarker right now for the PD-1 therapies, its correlation with efficacy is, I guess, fairly weak at best. It might enhance patients who do better, but there is a fair amount of efficacy for even patients who don't have high PD-L1 expression, especially depending on which measurement one uses. As it stands right now, does that affect how you view, using PD-1s at all? The reliability of the biomarkers, or would you still really, because their efficacy is so high or especially their durability for so many patients, even negative expressors, that you would just continue to ignore the biomarker and use it for everyone?
Yeah, that's a really good and important question, Margaret. So yeah, I agree with you. I don't think PD-L1 is a reliable or sufficient biomarker. And so I mentioned earlier that the immune response is complex and it unfolds over time. And so I think it's important to think of the immune response as a dynamic process. And it changes over time, but it also happens in different locations. So certainly the tumor site is a very important location to look at, and that's what everybody looks at now. But I would argue that may not be sufficient just to look at the tumor site. So, for instance, for my group, we're very interested in the tumor-draining lymph nodes, and I personally view that the lymph node is where much of the cancer-immune interactions occur, and so we're very interested in looking at biomarkers in these lymph nodes and potentially also in the blood. And then the other issue is time. So, I mean, if you're watching a football game and you only get one picture of the game, it's very hard to know what's happening in that game. So just looking at a single biopsy at a single time point, it's hard to know did PD-L1 ever get expressed in this tumor and it just went down, or it's about to come up later. So another issue that we work on is trying to get more than one time point to look at as well. So I would say this is just the beginning. So we're very interested in looking at more than one biomarker at more than one site and at more than one time point. I think that's when we can really understand what's happening.
Right, and I think there's a lot of panels being researched and more complex biomarkers being studied. We've gotten a couple questions in, Dr. Lee. I wanted to ask you, since you're talking about the toxicity with the combination, one of our questions was that for PD-1/L1 trials, patients with autoimmune diseases have typically been excluded. Will we see a new range of adverse events in real practice as these patients enter trials and are treated in real life?
Actually, I'm not sure I understand the question. So yes, the point is well taken that patients with pre-existing autoimmune diseases are excluded from these trials. What's the question again?
If those patients are treated, do you expect to see a new range of adverse events, or should those patients eventually be treated, or should they always be excluded from this class of drugs?
Oh man, that's a really good question. I would imagine that that since these agents are FDA approved, that there will be patients with pre-existing autoimmune diseases being treated with these agents, and we'll learn. It's very hard for me to predict, but it's important to keep in mind that even though anti-CTLA-4 antibodies and anti-PD-1 or anti-PD-L1 antibodies are all meant in the category of checkpoint blocking antibodies. They actually act potentially on different phases of the immune response, and potentially even at different sites. So CTLA-4 is primarily active during the early phase of the immune response, the activation expansion phase, and potentially actually more in the lymphoid organs like lymph nodes, whereas PD-1, PD-L1 is more prominent in the effector phase of the immune response and potentially in the tumor microenvironment. So this may be why we see more toxicity and autoimmune issues with anti-CTLA-4 blockade, because of the effects on priming and activation of the immune system.
And sort of picking up on that idea, I think a lot of the talk at ASCO was about how certain tumors can be cold or not immunologically active or hot, and about using different therapies like CTLA-4 to prime the cold tumors, to warn them or enhance the immune response. So Dr. Lee, what do you think about the potential to expand immuno-oncology interventions into additional tumor types or get better responses? And are there additional agents and interventions coming along that can help with that?
Yeah, that's a very important principle about, well, the notion that certain tumors are either immunologically hot or cold or inflamed and uninflamed, and that's certainly an observation that's, I think, partially valid. So we and others certainly find that there are human tumors that are infiltrated heavily by immune cells and others that are not. And there is some correlation between pre-existing immune cells in tumors and in response to checkpoint blockade. There's also some data to show at least some hints for why that might be. There's some work showing that the beta-catenin signaling pathway may have some role in whether or not tumors have pre-existing T-cell infiltration or not. So I think that's another area that we need to work on is how do you get more endogenous immune infiltration in the tumors and then combining that with immune modulation. So one concept that I think is very interesting that probably still not too many people know about is the notion of immunogenic cell death. So it turns out that cells can die in different ways and to the immune system. And apoptosis in general is immunologically silent to the immune system. And if you think about it, it makes sense because cells die in our bodies all the time, and you don't want an immune response every time a cell dies in your body. And so apoptosis in general is immunologically silent. But there are other ways in which cell dies, which does trigger an immune response. And some of those mechanisms are now worked out. And turns out that some chemotherapeutic agents, particularly doxorubicin, has been shown to actually induce immunogenic cell death of cancer cells. And so there may be ways to kill cancer cells in a preferentially immunoactive way. And so I think those are particularly promising ways to combine agents to combine with immune modulation.
Great. And I know a lot of, you've been also doing research into the tumor microenvironment. I think that's something, another area starting to see people looking at the interplay there. Is that something you could tell us a bit about?
Yeah, so for years, people have more or less thought of tumors as a homogeneous collection of cancer cells. So if you actually look at thousands of human tumors like we have, it turns out that most human tumors are only about 50% cancer cells. That's on average. The range can be as low as 20% to as high as 90% cancer cells. But the point is that tumors actually have lots of other cell types in addition to cancer cells. And why does this matter? So interestingly, there have been studies now in breast cancer, colon cancer, pancreatic cancer, almost all cancers looked at have shown a correlation that actually tumors with less cancer cells do worse than tumors with more cancer cells, which intuitively is paradoxical, right? You would think that tumors with more cancer cells should do worse. Yeah. And this really, I think, speaks to the notion that the microenvironment is actually very important in clinical outcome. And the other cells in the tumor microenvironment include immune cells, but also non-immune stromal cells, particularly cancer-associated fibroblasts. And so there now has been a lot of work showing that these cancer-associated fibroblasts play important roles not only in supporting the survival and growth of cancer cells, but they too modulate immune cells. For instance, when you actually look in high resolution at PD-L1 expression in a tumor, a lot of times PD-L1 is not expressed on cancer cells, or at least not only on cancer cells, but they could be expressed on stromal cells and very prominently on myeloid cells that infiltrate tumors. So they're a very interesting relationship between different cell populations in the tumor. So I think it's important. I think it would be useful to think of tumors now as organs and ecosystem. And so we're very interested in understanding interplay between the different cell types within the tumor microenvironment and take this into account in our development of new therapies.
Dr. Lee, this is Robert. So just to follow up, you've given us a lot of good information about, the sort of different mechanisms that we're understanding in terms of tumor immunogenicity. Are any of these likely to be potential biomarkers, for example, for selecting for patients who, outside of PD-L1 expression, are any of these likely to be useful biomarkers for selecting patients who either are likelier to respond to checkpoint inhibitors, or even as a measure of patients who are responding after treatment, before having to wait for a response?
Absolutely. So absolutely, I would argue that looking at the whole tumor microenvironment, the balance between myeloid cells and T cells, regulatory T cells versus CD8 T cells, what the cancer-associated fibroblasts might be doing. And then one other concept that we haven't gotten to yet is the notion of mutational load. I think these are all parameters that should be taken into account when we look at the bigger picture of what's actually going on and how to select patients.
Great. And I think that'll be very interesting to keep an eye on, especially sort of in different tumor types. It sounds like that the mutational load varies and could have an impact on the efficacy of immuno-oncology in those spaces. And I know you do a lot of research in breast cancer, and that's where Zachary has picked some slides. Sort of to move us into that direction, could you tell us a bit about whether or not there's potential for immuno-oncology in breast cancer?
Yeah, well, you're absolutely right. So this is what I primarily work on, is trying to develop immunotherapy for breast cancer. And as many of you know, the early trials in breast cancer with anti-PD-1 as a single agent has shown some response, but not dramatic. So in triple-negative breast cancer, the response rate to anti-PD-1 antibody as a single agent is somewhere in the order of 15 to 18%. So I think people have been not overly optimistic, but I think it's a start, and I would remind ourselves that even the early trials in melanoma wasn't that dramatically higher. And so the notion that response to checkpoint blockade is primarily driven by neoantigens and pre-existing T-cell infiltration of tumors, I think it's partially correct. And so there's a famous graph published in Nature a few years ago plotting the average mutational load of different human cancer types with melanoma, lung cancer on the far end of the high side, and breast cancer is pretty much in the middle. But if you look at human breast tumors, they too are infiltrated by T cells, and there's a strong correlation between T cell infiltration of breast tumors and better clinical outcome. So clearly the host immune system is engaged even in breast cancer. And we're starting to see that we can isolate the tumor-reactive T cells out of human breast tumors. So they seem to be targeting not just new antigens, but also non-mutated self-antigens. So I think we still have a lot to learn as far as what truly drives the immune response endogenously against cancer. But I am optimistic that once we understand these better, we will be successful in developing immune therapies for breast cancer and other cancers that are in the low mutational load category.
And that'll be very exciting to see. Zachary, you had picked out a couple abstracts about the CDK4/6 inhibitors in breast cancer. Can you tell us a bit about that mechanism and how these drugs fit into the breast cancer field?
Yeah, sure. So like Dr. Lee said, the immunotherapies haven't quite made their way to breast cancer like they have in other indications yet. So I think one of the more exciting mechanisms outside of that is CDK4/6. And just very, very briefly, CDK4 and 6 associate with cyclin D proteins, and they're involved in DNA replication and progression through the cell cycle. And inhibiting these CDKs when they're often overly expressed or activated in the pathway in cancerous tissues is a good way to fight cancer. So there's been some good data in this so far, obviously with Ibrance and palbociclib. And the first presentation I want to talk about is Paloma-2, phase 3 trial of palbociclib with letrozole compared with letrozole alone in postmenopausal women with ER-positive, HER2-negative advanced breast cancer. Ibrance has already received advanced conditional approval in this indication based on the phase 2 Paloma-1 trial for the first-line treatment of postmenopausal patients, and it's received that based on a 10-month PFS benefit over the standard of care letrozole in letrozole alone. So it's already received approval as well in combination with fulvestrant for breast cancer patients that have progressed on prior endocrine therapy as well. So in this trial, palbociclib was administered with letrozole every 28 days, 3 weeks on, 1 week off, in 666 postmenopausal patients with no prior systemic therapy. As you can see, median PFS was 24.8 months versus 14.5 with placebo plus letrozole. ORR was improved 42% versus 35%, and 55 versus 44% in patients with measurable disease. Clinical benefit rate was also pretty good with 85% in palbociclib versus 70% in the placebo group. So there were some serious AEs in about 20% of patients, versus only 12% of patients in the placebo arm, and there were some dose reductions as well. So basically, Paloma 2 trial was a confirmatory study for Paloma 1, and it reproduced a significant PFS benefit. Like I said before, Paloma 1 saw an increase in PFS of 10 months in the overall population, jumping from 10 months with letrozole alone to 20 months with Ibrance, and that was replicated here as well. And I'd like to point out that the PFS was increased from 19 to 30 months in a blinded central review. So there's obviously a benefit here with these CDK inhibitors. And this basically affirms Ibrance's position as a leading therapy in this indication and will lead to full approval in the U.S. and marketing applications in other markets. And this is a good timing as Ibrance needs to maintain its market share within this indication as Novartis's ribociclib and Eli Lilly's abemaciclib aren't far behind as well.
Thanks. I think you'd also picked out the Monarch 1 study of Lilly's drug. Can you tell us a bit about how this is stacking up as a competitor to Ibrance?
Yeah, absolutely. So for the Monarch 1 study, just a little background and context for abemaciclib. Many breast cancer patients will ultimately relapse or become resistant to NSAIs or endocrine therapies, and metastatic breast cancer patients that are refractory to endocrine therapies are recommended to undergo cytotoxic chemotherapy, and these regimens typically provide maybe 3 or 4 months progression-free survival, have several dose-limiting toxicities. So this study investigated abemaciclib in this hard-to-treat population. And as you can see in the results here, abemaciclib was administered orally on a continuous schedule in 132 patients, many of which had visceral metastases. They were heavily pretreated with a median of 3 lines of prior therapy. Confirmed ORR was 19.7%, clinical benefit rate 42%, PFS 6 months, and median overall survival was 17.7 months. The most common grade 3 AE was on-target toxicity. It was diarrhea, was 20%, and about half of the patients required at least 1 dose reduction. So I think in these heavily pretreated patients, abemaciclib achieves some significant single-agent activity, and they compare fairly well with Ibrance. The Paloma 3 trial of Ibrance stacks up pretty well as a combo of Ibrance and fulvestrant, where ORR was 10%, clinical benefit rate was 34%, and median PFS was 9.2 months. I will note that there were a large number of dose reductions. About half of the patients required a reduction from the 200 milligram starting dose. Dr. Dickler from the Memorial Sloan Kettering, who presented the data, mentioned that phase 3 trials, Monarch 3, is already investigating it as a combination with fulvestrant in a lower dose of 150 milligrams. So they're hoping that this efficacy does not reduce, but the lower dose will be more tolerable for these patients. And I think maybe the big takeaway from this is that even though there were dose reductions and adverse events, good efficacy was achieved with continuous dosing. This is in contrast to Ibrance, which requires dose interruptions, and this might be a distinguishing factor for abemaciclib in the future with its better tolerability profile. So we're waiting on data for the ongoing studies of abemaciclib as a component of a combination regimen, either with an NSAI or fulvestrant, as we think this might be how it's approved. So the market's large and growing, and we believe abemaciclib will have a pretty good chance up against Ibrance.
Great. Dr. Lee, since you're a breast cancer specialist, can you tell us your impression of the CDK4/6 class and how this new data will affect clinical practice?
Yeah, I think these data are very promising, and certainly CDK4/6 inhibition is definitely something that will be used clinically in the field. Actually, at City of Hope, we're working with a colleague to combine Palbo and letrozole with anti-PD-1 blockade as a triple combination. So I think there's really a lot of promise in this direction, and also a lot of interest in further enhancing these combinations. One thing that I would also bring up is there's actually some interesting data showing that the CDK4/6 inhibitors can also have some immune modulating effects. There's some publications on that, and so that's also quite interesting.
One of our participants has asked, and then either Dr. Lee or Zach, if you want to jump in, if you could comment on how efficacious the CDK4/6 inhibitors are in the first-line setting versus the second-line setting?
Yeah, sure. I will say that phase 3 trials seem to point towards probably being more efficacious in the frontline setting with aromatase inhibitors. You're looking at 20-month progression-free survival benefits. In the Paloma-3 trial of palbociclib and fulvestrant in relapsed patients who progressed on prior endocrine therapy, the PFS benefit was, I think it was about 9 months. So I think overall might be better off in earlier indications, or earlier lines of therapy, excuse me.
Great. So our final grouping is in lung cancer, where immuno-oncology is taking off, but Robert has selected some targeted therapies that may not be on everyone's radar. Robert, could you explain how you see other interventions fitting in alongside immuno-oncology?
Sure, and I'll be brief because I know we're winding down in the webinar. I basically picked two very related abstracts to talk about. Both of them are addressing ALK-positive lung cancer patients. So just for context, these are patients with a ALK mutation that are likely driving the disease, or at least part of the disease. And currently there are 3 approved therapies in this area for this subgroup. One, Xalkori, or known as crizotinib from Pfizer, is the only approved therapy for first-line treatment of ALK-positive patients. And then there are 2 additional ALK inhibitors, Zycadia, also known as ceritinib, and Elsensa, or alectinib from Roche. Those are both for use after progression on crizotinib, the first-line therapy. And so my first abstract was really, these are both pretty straightforward, so I'm not going to belabor the numbers here, but the first one was a presentation from the ALTA trial of brigatinib. And basically this is another ALK inhibitor, another second-generation ALK inhibitor that is aiming currently for second-line use after crizotinib progression as well. The ALTA trial is its pivotal trial, and you could see some of the details on this slide, but basically the data were pretty good, and it's a post-presentation discussant was pretty confident that it would get approved. I think there was a response rate around 50%, depending on which dose was used. It's going to go forward with the higher dose, but one of the interesting things from brigatinib was that it had pretty high efficacy in intracranial patients, so those with brain metastases, and that's something that I think is not seen with the currently approved crizotinib. Excuse me. So this is likely to be a new entrant into second-line therapy with the other ALK inhibitors. And then if we could go to the next slide, please. So then my next abstract is related. It's another ALK inhibitor. It's for alectinib. It's currently approved. It has accelerated approval for use after crizotinib progression, and it is being developed now for first-line use. And so this was a head-to-head trial. The J-ALEX trial was a Japanese trial that was comparing alectinib to crizotinib, the first-line therapy. And again, you can see the numbers here on the slide. I think that the key takeaway here is the PFS hazard ratio was 0.34, so alectinib was superior to crizotinib, and it was basically is likely to move into first-line therapy. There's another trial going on, an international trial that will be used for U.S. and European approval, not J-ALEX, but the ALEX trial. That's ongoing, and I think that's going to have a readout this year. So you expect alectinib to become a first-line therapy for ALK-positive lung cancer patients as well. And I'll just, that's all the detail I'll give about these 2 trials, but I'll just say that I chose this so that we could move this discussion, maybe away from immunotherapies for a second and think about how non-immunotherapies might still have a space in oncology treatment. And this seems like a clear demonstration of, or clear example of how they might move forward using a subgroup for subgroups or very targeted populations where we might have a pretty good understanding of the driver mutations or the mechanism behind the tumor, and so these will basically be highly efficacious for a small subgroup, and I think it's worth mentioning that along with that comes probably a very high price tag. So that's something that maybe we could wrap it up with and even discuss a little bit there.
Yeah, no, I think that would be great, because I think also both IO and targeted therapies can certainly cost a lot, especially if you're talking about combining or sequencing. And since value and drug pricing continue to be big topics at ASCO, I'd like to ask the panel for their take on what one of our listeners termed the dilemma between healthcare cost containment and the high cost of individualized therapies, which I imagine will only get worse with personalized drugs like CAR-Ts. So maybe if we could sort of go around and talk a bit about how the targeted therapies and immuno-oncology can fit together, and how cost is going to be a factor in these decisions. We could start with Dr. Lee, who has the most practical experience.
Well, but I probably have the least insight into the economics of it all. So I would mostly say that it's definitely a huge issue, especially when you're talking about producing personalized cellular therapies. So I think that is an issue that we have to face as a field and as a society. But otherwise, I will punt to the other panelists.
Well, maybe I could follow up and ask Dr. Lee a more specific question, and that is, in the two abstracts I just presented, or especially the second one, where we're talking about alectinib moving into first-line therapy, if the PFS of, and this is something that a post-presentation discussant was sort of highlighting, is that if the PFS of alectinib as first-line therapy was generally similar to crizotinib as first-line therapy followed by alectinib for patients who progressed after first-line therapy, would you factor cost in? So, if alectinib was significantly more expensive, would you factor cost in, or would the convenience or an imperative to use the most effective first-line therapy drive your decision-making?
Yeah, I appreciate this question, and I think it's treating physicians. I think there needs to be some criteria for treating physicians to select agents, and certainly if there are two agents that have equal efficacy side effect profiles, then cost has to be taken into consideration. So I would just say that I agree with you. I think maybe there should be some sort of algorithm by which treatment should be decided upon, factoring in cost.
Okay, thanks.
Yeah, I would add to that, going back to maybe the biomarkers and combinations of biotherapies, that we might see, the law of diminishing marginal returns on this, especially with the more extravagant IO combinations. As we try to get more creative with those therapies, there could be a lot more toxicity, and of course, all these combinations, the ever-present question of whether or not patients can afford these treatments. So I think, going back to the biomarkers to circle around as well will be an important point in finding the patients best suited to these therapies, especially when cost and toxicity are taken into account.
All right, and I think that's a fantastic point to end on. I'd like to thank you all for joining us today. If you have any further questions or comments, please email them to pharma@informa.com, up here on the screen. And thank you all again, and enjoy the rest of your day.